first thing I'm going to talk about is this image, and it should have been the first one that you drew. And it's a nice little flow chart, and it shows everything that happens from the very beginning of this process all the way to the ultimate final result. So everything we've been talking about is protein synthesis, transcription, and translation, and so that we make a protein. But the entire process has to get started somehow, and the signal typically, so this little tan area is your cell. The purple area is the nucleus. So everything inside the purple area is happening inside the nucleus. And then all of this yellowish stuff would be your cytosol, and we call it the cytoplasm because there's all kind of organelles in here. And then the signal, which is right here, is coming from outside the cell. And so whenever things need to get made in your body, hormone signal, different substances that are in your blood can create a signal, stress can create a signal, electrical impulses can create signals to release different ligands, and then they get received. So that was all in like chapter 6 was our cell signaling and signal transduction pathway. And this is the ultimate final end result of the signal transduction pathway. In the signal transduction pathway, the ligand is received on the membrane, and it's by a receptor protein, and then it creates this chain reaction, and then it finally gets to the nucleus. And when it gets to the nucleus, it stimulates or elicits a response. And everything we've been doing for the last couple of weeks is all about that response. So here is your chromatin, which is just your tangled up mass of DNA. And it's not in the chromosome form because this cell is not dividing. So the chromatin is all nice and opened up so that the messages in the DNA can actually be accessed. So now we have a little segment of that chromatin in the tiniest form that it can be found in, and this is your DNA. And right here it says this is that gene that it's trying to trigger to get this ultimate final cellular product. All right, so that gene gets triggered and transcription occurs. So transcription is when we go from a piece of DNA and we turn it into RNA. So the base pairs line up, but they're complementary. In DNA, there's A's, C's, G's, but there's T's for thymine. RNA, we still have A's, C's, and G's, and the base pairing rule still stands, except on the DNA, when we make our new piece of RNA, when A would typically bond with a T and pair up in DNA, A will pair up with the U in RNA. And this is just a temporary copy, okay? And that's huge. I want you to write that in the margin or on your picture there, that piece of messenger RNA is a temporary copy, photocopied section of this very, very important book. But within it right now, we notice that there is some segments called introns, some segments called exons. Now, one of the ways we know that we're dealing with eukaryotic DNA is that we actually have a nucleus. Prokaryotic cells do not have a nucleus, so none of this happens in the nucleus. It all happens in the cytosol. But eukaryotes like us, we have very complex cells, and so we want to make sure that this process is done very well. And so we keep it all in-house and do not release the actual piece of messenger RNA until we've processed it. And by processing, we remove our introns and we keep our exons. And then we're going to add our cap and our tail. So our cap is at the 5' end, and it's a bunch of G's, it's a bunch of guanines. And then the tail is a poly A tail, and so it's a bunch of adenines. And they don't code for anything. They're just there to keep the DNA safe. Now we're going to leave through the nucleus. The nucleus has little openings on it called pores. And those pores are very small, but a single strand of RNA is tiny enough to pass through. The double-stranded DNA is too big and cannot pass through, so it's the perfect size. So now our message is out in the cytoplasm, and that message is going to head for the ribosomes so that translation can occur. And the ribosomes are going to read that message in chunks of 3 called codons. transfer RNA is going to come along and it's going to bring all the amino acids, and you're going to get this long chain. It looks like a necklace. Now over here you notice this is the degradation of messenger RNA. So it's almost like a secret code in the war movies that you see. The guy brings the code and immediately he burns it, or he tears it into 1,000 pieces, or sometimes they even eat the message, okay? So the message gets degraded because if that message is floating around in this cell, every ribosome that finds it will just read the message and make the polypeptide. And if it makes too much of this, whatever this protein is, then your cell could be damaged, die. It could cause disease because you want to be like Goldilocks. You want to make proteins, but you only want to make them when you need them, and you don't want too many of them, but you certainly don't want to be missing certain proteins. So there's always this perfect balance, and in order to maintain this perfect balance, we degrade that message once it's been used. All right, so we have our polypeptide, and then we're going to process it. And this comes from chapter 2, where the protein starts to fold. And this is the primary structure. The secondary structure, it'll be alpha helix or beta pleated sheets. And then that will fold on top of itself. And then 3 or more of these polypeptides will cluster together, and now it becomes an active protein. So if you notice here, it does not say active, so it can't even do its job until it's folded correctly. All right, and then it either gets degraded after it's done its job. So once again, it's another scenario where if that protein is a temporary protein needed to do a very quick, fast job, but then get the heck out of there, then the cell degrades it. It can also be transported to other organelles in the cell, or it can actually be exported from the cell and secreted. So you get, these are enzymes or structure or things like that, okay? Now all of this process is all the stages of gene expression and all the places where we can regulate it. So we can regulate it here, we can not send a signal. We could regulate it here and remove different introns. We can regulate it, let's see, probably at this level by degrading it before anything ever gets made. So you could possibly get questions about how and where could gene regulation occur. And this is kind of a common sense question. Go with the stuff that makes sense early on, because if you never send the signal, then you never get the final product. If you remove the wrong introns, then you never get the same final product. If that RNA is blocked, if these pores have some sort of issue and nothing can leave, then you never get this final product. Biochemists work very hard for people who make too much of certain enzymes to somehow stop this process from occurring so they don't have all these overabundance of enzymes. On the flip side, biochemists also work for people who don't make them at all. They either try to give them the correct gene so that if they have the recipe, they can make it, which is biotechnology. So we'll take that gene and put it into somebody who doesn't have it, and if they have it, then they can make it. And the crazy thing is we could take that gene and put it in another organism, and another organism will actually make that exact same protein, because it's not a thinking process. It's a response. They get a signal, they trigger this gene, they make this, and then you have your protein. All right, so I'm going to stop the video here, and then we're going to move on to the next picture, which is the eukaryotic regulation of gene expression.